All right. The kind of official title of my talk today is Taste Representation and Processing in Drosophila higher brain. Basically, what I'm going to be talking about is how you can take a fruit fly, or Drosophila melanogaster, and you can use really cool genetic techniques and microscopy techniques to knock out parts of its brain, see what happens, and then watch what happens underneath the microscope. All right. So, how do we begin to understand complex behavior and decision making? Take something we do every single day. Okay, you guys, you walk into the cafeteria and you have to decide what you want to eat for lunch. Okay? Now, we have a menu. It has these helpful little dots next to it that I've only recently figured out what they mean. This is today's menu, by the way, in case anyone's interested. You walk in and you're bombarded with sensory stimuli, right? Sometimes you could smell the food, you can see the food, you can read about what's there, you can hear hundreds of teenagers speaking very, very loudly. You have your friends that are trying to influence your decision in some way. You know, maybe if you were there alone, you'd just be eating plain pasta like every single middle schooler does, because they don't have any shame. But no, you know, you guys are trying to make healthy, informed decisions. So you have this social impact, you have planning for the future, you have sensory impact. You know what things are going to taste like, what the texture is going to be like, what they look like. Maybe the broccoli looks a little off today. And then also, you know that you have memories of all these things, right? So this isn't, you know, going to be the first time, you know, you've ever eaten scrambled eggs, for example. So you have a memory of what scrambled eggs are like. You look through the cafeteria and you see certain visual stimuli or have certain odor stimuli that are going to trigger a memory. You see a big glass bowl out on the table and you think, maybe today's the day for an ice cream novelty. Okay? But then you remember, oh, the last time I ate this strangely shaped ice cream cone, I had a bit of a stomach ache. So maybe you don't want to do that again. So all these memories, even something like just deciding, should I eat this or should I not eat this, all of these other senses are involved, and memory comes into play as well. So how am I actually going to understand all the different things that are going on in your brain that help you make a decision? And one of the ways we do that is that we can use what are called model systems. Okay? And so instead of taking your very complex human brains that are trying to process all of this information with billions of neurons, what we use are smaller animals that have simpler brains and allow us to look more directly and specifically at their brain cells, at their neurons. Okay? So here's a picture. Here's a human brain next to a mouse brain, next to a fly brain. Okay? Now, you can see there's definitely a difference in size, right? Obviously, this brain is not fitting into a fruit fly, right? Okay. And you can see, you would probably guess there's also a difference in complexity, okay? So, instead of having billions of neurons, fruit flies, the system I worked with, has about 100,000. So that's still a lot. Fruit flies can still do pretty complex behavior. They can decide what to eat, they can decide who to mate with, where to lay eggs, all these kinds of things. They do all these different behaviors. They have memories, which we're going to be talking about today. But it's in a much simpler system, and because it's a fly, we can play with it a lot more, right? I can control its genetics, I can control its environment, and I can look directly into its brain, which is what we're going to be talking about today. Now, the fly brain is so small that whoever made this graphic didn't even bother putting an actual fly brain on there. They just put a little white pixel. But I'm going to zoom in with you guys here. So this is a picture of the fly brain, obviously magnified. It's a little bit cut off here. These are actually the visual lobes, and I'm going to introduce you to a couple of different regions that I'm going to be talking about today. So it looks obviously very different from a human brain. Invertebrate brains are organized differently, but they do a lot of the same functions, which lets us ask some of the same questions and look at how information is being processed, even in this different system, is processed largely in a lot of the same ways. Okay. Okay. So first, Hello. Okay, so first is this region here, which which I'm going to be talking about, called the subesophageal zone. Another weird thing about invertebrate brains, this fly brain in particular, this right there is called the esophageal opening. Anyone want to guess what that means? The esophagus actually runs through the middle of this guy's brain. Okay, so this region underneath is called subesophageal zone, and this is where a lot of taste information comes in. It's the primary sensory relay for information coming from taste neurons. All right. These are the antennal lobes. So flies sense odor with their antenna, and that information comes in to this area right here, the antennal lobes. And then there's another region we're going to be talking about today. Skip the lateral horn. We're going to be talking about a region called the mushroom body. Okay? And these regions are very, very important for learning and memory in the fruit fly. So they've been linked to all the different kinds of memories you guys probably think of. Fruit flies are pretty simple. So most of their memories are, okay, I remember that visual stimulus is bad or good, or I remember that that smell is, if I smell that smell, I'm going to get an electric shock, something like that. In my case, I'm going to teach them that sugar is bad, okay, briefly. Okay, so this area of the mushroom body I'm going to be talking about mostly today. And this region has largely only been studied in olfaction, so in smell. And I wanted to look at how it is also involved in taste information, okay, or gustatory information. It's a fancy way to say that. All right. Now, the mushroom body, as I mentioned, is a center for specifically a type of learning called associative learning. So this is when you take, so if you guys have all learned about Pavlov's dogs, right? They heard the bell and they would drool because they associated it with food. This is a similar kind of region, a region that does similar kind of processing. So if a fly, so we know mostly about olfactory memory. So if a fly smells a specific odor, and then you pair that odor with some kind of stimulus, a positive stimulus or a negative stimulus, this will allow the fly to learn that this odor is good or bad, has a positive or negative valence, is the way we can say that. So the odor, as I said, it goes to the antennal lobe, it gets processed here. And then these neurons, they look like this. So again, a neuron is just a brain cell, okay? So it's single cell, like you're learning about in Bio 1, Bio 2, okay? But they have they have this cool morphology. They have these cool shapes where they can extend out really far. So here's just a cartoon of a single neuron here that is what we call a second order olfactory neuron. And it projects up to this structure called the mushroom body, and it talks to the neurons in the mushroom body. And the mushroom body is where this pairing takes place. So if I pair an odor with a negative stimulus like shock, or with an appetitive stimulus like sugar, something good, the fly will learn that this odor is good or bad. And this happens in the mushroom body. The mushroom body needs to be there. If it's not there, they then can't learn this association. If it is there and these pairings happen, then you get what either we call aversive learning, so avoidance learning, they learn that that's bad, or appetitive learning, they learn that that's good. Okay. So I should change this a little bit. So I looked at the neural basis of aversive taste learning, and specifically wanted to look at what is the role of the mushroom body in aversive taste learning. Which neurons convey that aversive reinforcement signal, so that shock signal, what neurons are doing that in this case. And then overall, how is taste represented? So how are the neurons in the mushroom body actually firing, actually communicating with each other that is sending and receiving taste information? Okay, we're going to skip that part of the project because we don't have time. Okay, here's a fruit fly called Drosophila. Okay, obviously zoomed in. You see these guys in your kitchen all the time. Now, a fruit fly is a little bit different from us when it comes to taste. It can taste with its mouth parts. Its mouth is called a proboscis. And when it senses food, it will stick its proboscis out and it acts basically like a straw. It can suck up its food. But a fly tastes not only with its mouth, but also with its legs or tarsi and its wings. So when a fly is walking around on your skin, it's actually tasting your skin. Okay? And that way, it's not that bad. Okay, and that way, it makes no difference to you, just to the fly. Okay, so, and that way it can taste its environment and it can decide, oh, okay, I think I'm walking around on a rotting banana. That seems awesome. So I'm going to extend my proboscis and I'm going to eat. Okay? All right. So I designed a very simple paradigm here that I'm going to show you guys a quick video of. So I would take flies and I would deprive them of food for a little while, so they'd be really hungry, really motivated. It'd be like when you're waiting for third lunch. It's really bad. Just can't make it through that class. Okay. So so these flies right now, I have it kind of glued down, and you're going to see me. I'm going to present a little drop of sugar to the fly, and it's going to touch it on its legs. And because it can taste on its legs, it's going to sense, oh, this is good sugar, and it's going to extend its proboscis ready to eat. Okay. Then I'm going to test flies to make sure that they're doing that properly, but then I'm going to trick the fly, okay? So I'm going to give the fly sugar on its legs, but then when it extends its proboscis, I'm going to swoop in with a really, really bitter substance. So it's going to think, I'm going to drink sugar, and then what it's actually going to drink is something called quinine. It's very bitter, okay? And then once the fly has that kind of pairing and it's learned that information, it's going to, when it tastes sugar, it's going to say, no thank you, right? It's going to avoid it, and we can tell that because it's not going to extend its proboscis to drink, okay? So trial, you pair it, and then the test is you present the sucrose alone, and you look for, extension. Do I not actually have the video? Sorry guys, again, grad school computer, it's taped together. Okay, stop, okay. So the video didn't work, but you can get the idea here from this picture. So you present the fly with sucrose, you train it with the bitter substance, and after that, it will choose not to extend its proboscis. Here you can see a graph of what this learning looks like. So up here would be full extension, so this would be the fly extends all the time. If the fly is not trained, it will just always extend to the sugar, okay? If the fly has been trained, so here's a single pairing right there, if the fly has been trained, the majority of flies, so this is the percentage of the population that's not extending, the majority of the flies will not extend their proboscis for something like 15 to 20 minutes. And then they'll forget and they'll go back to extending the proboscis, okay? So I won't get into the details of this, but the mushroom body has a lot of distinct pieces, and each of these pieces do specific things. So one thing I wanted to do was see which of these pieces was important. And to do this, I used a cool genetic technique called conditional silencing. And so because we have basically full control over the genetics of these animals, we can do really neat things. And one of these things is I can put in a silencer in a section in just the neurons that I'm interested in. I can put in this special ion channel. And at normal temperatures, the ion channel is not going to do anything, so the neurons can function totally normally. But then if I move the fly to a slightly hotter room, what's going to happen is that ion channel is going to open up, and then the neurons are going to be unable to fire. So they're going to be unable to talk to each other and send information. So basically what I've done is I've erased that part of the brain for a few minutes, right? That part of the brain can no longer work. And so I'm going to do that. I'm going to shut off different parts of the brain and then see what happens, okay, to my learning. So I did that, and I won't drag you through everything. But basically, I was able to show which parts of the mushroom body were important. You can see here, for example, this is maybe the easiest graph to interpret real fast, that if the fly was not at hot temperatures, it could learn. But if it was at hot temperatures, so if that part of the brain was silenced, it could no longer learn. And so this told me a lot about which neurons were actually important for this behavior. I'm actually going to skip through this part, but because I want to get to the real fun stuff. Then the main thing, the main part of my study was I wanted to actually look at what was happening under a microscope. Okay, and to do this, we use a really cool, other cool genetic technique, and this one's called GCAM. And so this right here is GFP. Does anyone remember from class or anything what that stands for? You might have learned this in research or something.
Fluorescent protein.
Green fluorescent protein, yes, green fluorescent protein. So this protein normally lights up bright green, okay? But what scientists have done is we've modified this and we put a calcium sensor on it. So it's only going to light up when there's calcium around. And the reason we do that is because calcium will flood into a neuron when it fires, when it talks to another neuron. And so this way, this protein, it's going to be dim, but then if a neuron, and it's in the neuron, but then when it fires, all this calcium comes in, the calcium binds and it changes shape, and then it glows bright green. So then we can actually look at this and see which neurons are active, which neurons are talking to each other in real time. And skip this. Okay. And then the other neat thing about this is that I'm using a very cool microscope that allows me to actually image, we call it in 4 dimensions. So what this means is I'm actually taking, I'm looking at basically a 3-dimensional cube. I'm imaging through it really, really quickly. And then I'm looking at that over time. So what I can do is I can actually take the fly and I can look at a whole area. In my case, the mushroom body. I can look at the whole mushroom body. And I can watch what happens over time. And when I see things light up, when I see things fluoresce, I know that they're active. And I can say, okay, whatever I just did to the fly has made those neurons active. Okay? I wanted to quickly show you what that data looks like, except it is no longer open and this computer's terrible, so I won't do that. Okay. Anyway, so actually what I get is this really cool video. I didn't have the ability here to actually show you the kind of 3-dimensional rendering of it, but you can watch this whole volume light up over time. And then what I would do is I would look at it. I would look at what I was presenting the fly with. In this case, the fly is under the microscope, it's perfectly happy, except that it has a hole in its head. Okay, so I've attached the fly like this. I've cut a little hole in the back of its head and exposed the part of its brain that I'm interested in. Okay, and then I use this microscope to look at the brain. And what I can do is I can present the fly with some kind of stimulation, in my case sugar or bitter or something like that, and I can watch what happens. And particularly, I'm looking at this region of the mushroom body where the olfactory neurons are talking to the mushroom body, okay? And so those of you who learned a little bit about neurons, basically they have one side that listens to information, that's called the dendrites, and they have another side that passes on the information, that's the axon. So I'm specifically imaging individual dendrites, okay? So I'm looking specifically at one place where one neuron is talking to a bunch of other neurons, and I'm looking for whether it lights up or not, okay? And so, so as I said, I would go through this kind of whole area of this mushroom body, and the first thing was just to see, are these even going to light up with taste information? Because this structure was only really studied with olfaction, with smell, and so the question was, does taste information even come here? And so this is just a slice through this part of the brain, so it's a little bit hard to see here, but in black and white is a slice through this part of the brain that I'm looking at, and everything that's in red is something that fluoresced, is something that lit up, and I can see that I can see this repeatably, okay? So if I continue to apply sucrose to the fly, I get these very nice specific jumps in intensity. And because I get these nice jumps in fluorescent intensity, I can say, okay, that means there's more calcium. Because there's more calcium, that means the neuron is firing. And you can actually see these nice little rings here. These are called claws. And this is actually the dendrites that are wrapped around part of the axon of the olfactory neuron. So then what I was able to do is I was able to go through and I was to do some comparisons. So we didn't even know this structure listened to taste information, okay? But I was able to say, okay, we know that it does now, so what does it care about? Does it care if it's sweet or bitter? Does it care if it's a really intense sweet or a really low concentration sweet? Does it care if it's coming from its leg or if it's coming from its proboscis? And I was able to do these comparisons. So in the same fly, I could look at what parts of the brain lit up with sugar, what parts lit up with bitter, and how it compared. So you can see here, if you look here, so the green would be dendrites that were responsive to sugar, the red to bitter, and so you can see there's very little overlap. So it seems that these two streams of information are kept totally separate, even up here into the higher brain. And a similar kind of story for another kind of taste, water taste. I don't see much overlap. But what about different kinds of sugars or different kinds of bitters? There's a lot of different types of sugars or different types of bitters. Does the fly care about that? Is it able to distinguish one sugar from another at this stage of the brain? If it was, you'd guess that they'd be represented differently. But in this case, what you see is if I use sugars that taste the same level of sweet to the fly, or bitters that taste the same level of bitter, I see the exact same representation. So this means the fly probably can't distinguish between one sugar and another sugar, or one bitter and another. And I went back and did these kinds of same behavioral studies, and I found out that that's actually the case. I did a similar kind of thing looking at legs versus proboscis, or one side of the body looking at the other side of the body. And I was able to tell a little bit about how the circuit looked from this. And then another important thing is that taste information and smell information are very much linked in the fruit fly and in us, right? Our taste system is actually pretty limited, but we get this wide range of flavors because of smell, because of vision, because of touch, because of texture. And so at some point in the brain, these senses have to come together and integrate so we can tie them up together and compare them. But we don't really know exactly where this happens, and in the fruit fly brain, we didn't know where this happened either. And so one of the things I discovered here is that this seems to be one of the first places where information from both sensory systems is coming together. And this would allow for representations, for example, having an odor representation and a smell representation in the same place would allow you to, for example, learn a connection between the two things. So this would be an important area of the brain for further study. And so just to kind of summarize, I looked at where in the brain this was important, how it's represented, and then with that, I just wanted to point out, so I worked at a lab in UC Berkeley. If you guys ever have any questions about graduate school or about Berkeley in particular, feel free to come talk to me. These were my lab mates climbing a tree for some reason. That was my boss, Kristen Scott. And so I just wanted to thank you, send out a thank you to them. Obviously, they were integral in all of this kind of work. And then to ask you guys if you have any questions. We have like a minute. Anyone have a question?
How many pairings were required in order to achieve the learning?
That's a good question. So they can actually learn in a single pairing. It's just a very short memory. If I do up to 20 pairings, they'll remember that for about 2 hours. One pairing will be about 10 minutes. Anything else?
How did you attach the fly to the bottom half?
To the scope?
Yeah.
To be honest, I took a petri dish and I drilled a hole in it and I put some packing tape over it, and then I cut this really, really small piece of packing tape that fit between the fly's head and the body, and I held it in like that, and I cut a little hole in the head. In graduate school, you learn to use whatever you have. So there's a lot of things like drilling holes into petri dishes and using tiny pieces of packing tape. All right, thank you guys for coming.